All right. So the outline, I'm just going to walk you through the current hypothesis and limitation of observational studies and the new technique to address some of the biases that you've read a lot about, you've heard a lot about, and some caveat of the new methodology. So, in concluding remarks. So my talk will be focusing on more the method on is the relation between moderate drinking and health still valid in terms of what kind of bias can we address or are we still able to address? So what is the current hypothesis from observational study about moderate drinking and health? So you've read and heard about the virtue of moderate drinking in terms of cardiovascular disease, in particular myocardial infarction. So the biology is strong from increased HDL cholesterol from small to medium-sized randomized trial to platelet aggregation and fibrinolytic activity of moderate drinking. So here in this slide, the meta-analysis of reported study looking at coronary heart disease mortality show a 25% lower risk comparing moderate drinker to non-drinkers. So if you look at beer in particular with respect to fatal, non-fatal cardiovascular events, since this is a beer symposium, try to find some few slides that target beer in particular. You see that in a moderate range that you have some benefit somewhere from 20 to up to 40% lower risk. So peripheral arterial disease, here again, beer, moderate beer consumption is associated with a lower risk. These are data from the Framingham heart study. So heart failure, this meta-analysis shown by cubic spline. You can see also that in moderate range, whether using US, Canadian, UK guidelines, it's supporting it. But does that mean that all studies are showing the J-shaped relation between moderate consumption and all-cause mortality? The answer is no. And you've read this paper by Stockwell and colleague in 2016. Some of the argument that we'll talk about briefly, but for the sake of time, just going to be limiting maybe to 2 or 3. The argument that the abstainer bias, that some study did not separate a lifetime abstainer from say quitter or former drinkers, can be debunked by other well-randomized, not randomized, but well-conducted study. Here's another study where the relation between moderate consumption and all-cause mortality, sorry about that. So the multivariable adjusted, if you look at 7 to 13 drinks per week, there's no relation, no beneficial effect as expected based on the J-shape. So the question is, are we asking the right question? Is the choice of the outcome relevant? Because if we answer the wrong question, of course, we're going to saw inconsistency in the literature. So here, in this meta-analysis that I show you by Stockwell, the outcome was all-cause mortality. In particular, they excluded CHD-related death. So if you look at this analysis by Rohrkeg and colleague, they use lifetime abstainer as a reference group. And you can see here that down below, you have a J-shape looking at CHD death. Former drinker, they have a higher risk. And here, the pattern matters because from Russian subject where heavy drinking and binge drinking is frequent, you do not see any benefit, consistent with what Helena just discussed a few minutes ago. So, and it's obvious also here that if you separate episodic heavy drinkers, there's no benefit, actually there's harm. So if you binge drink, there's harm, but if you drink moderately, frequently, and compared to lifestyle abstainer, there is some protection. So the question becomes, is the choice of all-cause mortality a distraction rather than a scientific question that's based on the pathophysiology or the physiology of ethanol on a key intermediate factor? So if you're lumping outcome for which we know that there's definitely increased risk, such as breast cancer, where those lines show a linear and positive relation, or oropharyngeal cancer, colorectal cancer together, and the other one would be hemorrhagic stroke, where we know any amount of alcohol may be associated with an increased risk of hemorrhagic stroke, not ischemic stroke. Then you kind of throwing the baby out with the bathwater. In that, you're mixing the positive effect with the negative effect and biasing the results towards no effect at the end of the day. So while digesting the literature, we have to be asking ourselves the question, are we asking the right question, or are we reading the answer to the right question? So limitation of observational study, you are aware of them, including in particular unmeasured confounding. The one that we measure, we can deal with it to a certain extent, but things that we can't measure, we can't collect information on, it's hard to adjust for. Reverse causation, we can use a lifetime abstainer. Information bias, things like social drinking, some culture is not acceptable, so participant would not tell you always the truth. They may underestimate what they're consuming, or they may not recall accurately despite their due diligence. Misclassification, and so forth and so forth. So the new technique called Mendelian randomization is trying to avoid some of the caveat of the observational studies by using instrumental variables. So what is the goal of Mendelian randomization is to minimize bias in observational study where randomization is not possible or unethical. So it takes advantage of a random assortment of chromosomes during meiosis, gamete production, and use the genetic loci as instrumental variable, such as alcohol, metabolizing gene. So what is an instrumental variable? So suppose we want to study the relation between alcohol and heart attack. So alcohol will be the exposure, heart attack will be the outcome. So then we can use a genetic loci that metabolize alcohol as instrument. So the condition is that we're not allowing any connection between the instrumental variable and the outcome unless it goes through our exposure of interest. If that assumption is violated, then we're done. Then the result will be biased. So the two key assumptions that should be considered. Number one, is the instrumental variable a predictor of the exposure? What's the correlation between the genetic locus or genetic loci and alcohol? How much of the variance of the alcohol consumption is explained by genetic makeup? And the exogeneity in that the instrument cannot influence the outcome by other means other than the exposure of interest. So, but all these assumptions always match in published data. Failure to meet exogeneity, meaning if there's some pathway by which the instrumental variable affects the outcome, not through the exposure, then we're going to have a higher bias that we can measure because it's unobserved. We don't even know what pathway, they have not been discovered yet. So that's a problem. Second, the instrumental variable is a weak instrument, meaning it doesn't explain a lot of the variability of the exposure of interest. So, and even if those two conditions are met, Mendelian randomization is a large sample size technique, so it requires large sample size. So, conduct small study using the technique are more likely going to be biased. So here, a Venn diagram to explain what I just said. So if you consider the blue egg here, alcohol intake, the yellow would be cardiovascular disease, and the genetic locus, let's say alcohol dehydrogenase or 3 or acetaldehyde dehydrogenase 2, then you can see that what we're after is how much of the heart attack is explained by moderate drinking, the intersection of the blue and yellow. So because we can't randomize, we're using the gene that metabolizes alcohol, let's say ADH3, but ask ourselves how much of the alcohol drinking is explained by that genetic marker. If it's very little, then we have a problem. So here's another example where it explained a little bit 40% rather than less than 10% here. So the blue circle explained a lot by the purple, so that's good. It means we have a strong instrument, but it explained only a little bit of the effect of the exposure. So the best scenario would be to have an instrument that explains over 80% of the exposure and also explain a lot of the overlap, meaning the effect of our exposure are explained by the genetic variability. So a more realistic scenario is we have a non-dominant locus. Traits are affected by multiple genes now in the area of omics, where we have the whole DNA sequence. We can do GWASs. So we have, if we have one marker, we may explain maybe 2%, 3%, luckily 20% of the variability of our exposure. And it's an uphill battle to try to take that little amount to explain the larger effect. And if the effect of exposure is small, then it's even harder. So this helps explain the inconsistency of report that you read in the literature, because people say, well, researchers, are they crazy or what? Today it's protective, tomorrow the other people say, well, it's not. So which one is which? So here's an example of alcohol metabolism with a two genetic enzyme driven by genetic loci that we can measure, alcohol dehydrogenase 3 and aldehyde dehydrogenase 2. So a mutation in these enzymes over here can lead to slow metabolizer, which means more alcohol in the bloodstream. And over here, a point mutation to ALDH2 would lead to accumulation of this byproduct that has been shown to be carcinogenic. So what happened is, if we anticipate that beer would protect against coronary heart disease, then we would expect that the slow metabolizer would have a high amount of alcohol in the bloodstream and therefore have more benefit. So that's the principle of Mendelian randomization, that the genotype homozygous for the gamma-2 allele slow metabolizer would have a larger effect compared to the Y-type gamma-1, gamma-1. Is there evidence of such relation? Yes. If you look at Heinz paper looking at ADH3 and risk of heart attack, you can see that the homozygous slow metabolizer, they have a 35% lower risk of developing heart attack compared to the Y-type and the heterozygous intermediate. So we look at the cancer with acetaldehyde concentration. Here is that if you are slow metabolizer of acetaldehyde, accumulation of this toxic product could lead to oropharyngeal cancer, including esophageal cancer. And also a mutation leads to facial flushing, nausea, headache, and drowsiness, therefore leading to avoidance of alcohol for people with that mutation. So if this were true, then we will observe that people carrying these two alleles would have a lower risk of esophageal cancer. Is that the case? So this particular study looking at just esophageal cancer, comparing homozygous for ALDH2 versus 1-1, is showing 64% lower risk of esophageal cancer. So the proof of concept is demonstrated, but I wish it would work like that in real-life situation. So some of the caveat of Mendelian randomization, population stratification is that different group of people have different frequency of the genotype and disease frequency. And you can fix that by doing stratified analysis. Linkage disequilibrium is that some genetic marker that is too close in physical distance, they get inherited together. So we may be thinking about this locus, but indeed the actual genetic marker is somewhere else, close by. Genetic generalization is that our inability to infer the genotype from the phenotype. So nature is smarter than we think. The penetrance, genetic penetrance, that when you have the genetic locus, are you going to express what is expected? So you have 100% penetrance, yes, mostly dominant, even Huntington's disease, it's close by, but not quite 100% in some area. So what are the future directions? So in the era of omics, it's too simplistic to take one locus. So because we know the interconnection, you got to take advantage of the genetic loci that are discovered through GWASs, etc., and use what's called multivariable Mendelian randomization. I'm not going to go in detail to that. And while awaiting the first large trial of alcohol and cardiovascular disease, one's underway, started last year, recruiting about 8,050 plus age men and women across the globe. In 6 years, we will learn randomizing 14 grams of alcohol versus abstainer. So in conclusion, the appropriate instrumental variable with the appropriate sample size can lead to causal estimation effects of moderate drinking on health. So violation of the assumption is something to consider in terms of validity of the study. And as far as the J-shaped relation with coronary heart disease and other conditions like diabetes, the scientific evidence has not changed. It's just how we answer the right question. I'll stop here and thank you.